the acidity inside the cell. That reduction in acidity does the following things. Number one, this fusion of the virus envelope with our membrane to then allow its RNA to arrive into the cell, this fusion is hindered. This fusion requires acidic environment inside the cell. And in the presence of hydroxychloroquine, because the environment inside the cell has become less acidic, the fusion becomes hindered. So that is one mechanism. Second mechanism is, and I want to make sure that we are aware of this, that it's not just that the hydroxychloroquine is a zinc ionophore. These are extra functions or mechanisms by which it helps. So the second mechanism is that when the hydroxychloroquine is present and the environment is alkaline, now the virus has an enzyme called RDRP, RNA-dependent RNA polymerase. This enzyme is the one that would help the virus make its copies. And alkaline environment is bad for this enzyme as well. All of the cellular machinery needs acidic environment. So the second issue that occurs for coronavirus in the presence of HCQ is that the RDRP cannot function correctly. And that is another mechanism by which hydroxychloroquine stalls or slows down the virus replication. Number three, the inside of these lysosomes and the Golgi apparatuses or the endosomes, the machineries, inside there the virus has its particles that are generated. So remember, viruses they cannot normally divide. They're not living cells with the division and the nucleus machinery. They are RNAs. So they need to be, their small components are made separately, just like a car is manufactured in separate components and then we put them together. Similarly, the virus components are made separately in the cell and then they are put together in the Golgi apparatuses. And Golgi apparatuses would also require acidic environment. And when the hydroxychloroquine is present, the Golgi apparatus and the endoplasms cannot work correctly. And the result is so they cannot work. Sorry, I was looking at the comments. There was some comment about the video. So now that the endoplasmic reticulums are not able to work correctly, that means the assembly of the virus cannot occur correctly as well, or cannot occur. So virus assembly is stalled as well. So that is the third mechanism. A fourth mechanism is that hydroxychloroquine acts as a zinc ionophore. Zinc is a charged particle. It has 2+ charges on it. And charged particles cannot cross our cell membranes and go into the cell because our cell membrane is neutral and it would not allow the charged particles to cross from here. So we need some sort of a channel through which the zinc can go in. And if the zinc enters the cell, then the zinc can attack the virus enzyme RDRP as well and stall its function, which is sort of an antiviral effect of zinc. But this effect would only occur if zinc can enter the cell. So hydroxychloroquine, the next mechanism of action is that hydroxychloroquine would act as a zinc ionophore. And once it acts as a zinc ionophore, it gets inserted in the cell membranes as a channel and allows the zinc to come into the cell. And once the zinc is inside, it would go independent of the hydroxychloroquine and work on RDRP enzyme and stall the viral replication. A fifth mechanism, which is also very interesting, I love this mechanism. What happens is that these ACE2 enzymes, when these are produced, remember the ACE2 enzymes we just talked about? These are the enzymes that act as a receptor for the coronavirus. When these enzymes are being formed inside the cell, I call them, we give these enzymes a candy to then go and do their function. So when these enzymes are formed, the last amino acid on the enzyme needs glycation. What does that mean? That means that we take a glucose molecule and we attach it to the ACE2 receptor or the enzyme, and that completes the enzyme. Hydroxychloroquine interferes with the last amino acid glycation of the ACE2 receptors. The result of that is that when the ACE2 appears on a cell's surface, it has a slightly different shape compared to the regular ACE2 enzymes, because one glycated amino acid is not glycated. The result of that is that when coronavirus S protein tries to bind, so let's say this is the coronavirus again here, and the S protein is here, when it tries to bind with this modified ACE2, the affinity of binding is not strong enough and it kind of slips out. So that is the fifth mechanism. So if you now look at it, on one end, hydroxychloroquine would not allow an efficient binding of the coronavirus with our cells. Secondly, it would not allow the coronavirus to fuse with our cell membrane to allow its cargo, that is RNA, to enter the cell. Thirdly, if the RNA has entered the cell, the hydroxychloroquine has made the whole environment less acidic, which stalls the cell's activity. Then it is a zinc ionophore. So these are the mechanisms by which hydroxychloroquine stalls and stops the replication of the virus. And this is how it slows down the virus. And this is why it is also important that hydroxychloroquine is and grow. So this is the discussion that I have for you today.
Thank you so much, Dr. Bean. I think it's so important that we have experts like Dr. Bean, because most average physicians, myself included, cannot remember all of that. But the important point for the American people is that there are amazing scientists out there who inform our decisions. And then we're here to tell you what happens in the real world, knowing that there's basic science to back all of that up. Dr. Urzo.